frontier in. So here we are, and we have our esteemed guest of panelists, and I hereby welcome all the participants on this in this webinar today. And this webinar is hosted by CHIP Foundation in association with ECHO India and ICGA, that is Indian Cancer Genomic Atlas. So I hereby welcome the session moderator, Dr. Ankita Singh. Hi, Dr. Ankita.
Hi, Dr. Shekhar. Thank you so much.
Dr. Ankita is the head of scientific affairs at the Indian Cancer Genomic Atlas Foundation, where she leads scientific planning, collaborations, and programs development. She holds a PhD in cancer biology from the Institute of Genomics and Integrative Biology. Following her doctoral research, she pursued postdoctoral work at the Indian Council of Medical Research and the Council of Scientific and Industrial Research, where she transitioned from fundamental cancer biology to working extensively with clinical samples. Her expertise lies at the interface of cancer genomics and translational research with a focus on bridging discovery science and clinical application. With this background, I hereby welcome Dr. Ankita to this platform, and I would request her to take over the podium and I request her to please introduce our esteemed panelists and proceed with the webinar. Thank you.
Thank you so much, Dr. Shekhar, for this opportunity. And I thank the ECHO platform and the CHIP Foundation for collaborating with us and working together to present this webinar series to all our audiences. So briefly, the Indian Cancer Genome Atlas Foundation has been working to create a comprehensive cancer genomics reference for India, enabling collaborative research that can truly transform cancer care for our Indian population. Through this webinar series, we bring together experts who share their perspectives on frontier areas in cancer research and its clinical translation. So today's session is focused on multi-omics approaches in cancer, the next frontier in precision medicine. By integrating genomics, transcriptomics, proteomics, and metabolomics, multi-omics is showing a tremendous promise for earlier detection, better prognosis, and personalized treatment, especially for Indian patients where cancer biology is diverse and often presents unique challenges. Here, I take the pleasure to introduce both our speakers for the day, Professor Vijayalakshmi Ramshankar and Dr. Gagandeep. Our first speaker, I would like to invite Professor Vijayalakshmi, who is the professor and head of oral oncology at the Cancer Institute, WIA, from Chennai. She is a leading cancer researcher and molecular oncologist with a strong focus on the genetic and epigenetic mechanisms which underlie cancers, especially in context of Indian patients. Professor Vijayalakshmi has been deeply engaged in several collaborations, including with ICGA, and has played an instrumental role in advancing precision oncology in India. Her talk today would be centered around the clinical and diagnostic applications of multi-omics in Indian cancer patients. Thank you, Vijay, for joining us today, taking time for us today. Thank you so much.
Thank you, Ankita, for your kind words. So if I'm allowed, I'll start sharing my screen.
Sure, Vijay, yeah.
So is my screen visible?
Yes, we can see.
Okay. See, at the outset, I thank ICGA Foundation for this wonderful opportunity to share my views on multi-omics approaches in cancer, which is the next frontier in clinical medicine, precision medicine. And we are basically based out of Chennai. I head the Department of Cancer Biology and Molecular Diagnostics, which caters towards cancer research as well as molecular diagnostics for the hospital patients. So when we get an opportunity to do molecular diagnostics on the clinical samples, we tend to generate wealth of data. So that actually helps us to ask important clinical questions which can be shaped up into research grants. So that way I have immensely benefited out of molecular diagnostic exercise, because one way we are trying to be relevant in the hospital, giving relevant molecular diagnostic results to the patients based on which the treatment is happening. On the other side, we also tend to look at the data that we generate in the real world out of our samples, and we can ask good research related questions. So is my slide moving?
Yes, Vijay, you can put it on full screen mode. Yeah.
Yeah, one second. Okay. So multi means many, and omics is also something which is related to large scale. So in this multi-omics, we are all living in the era of multi-omics where we deal with integration of data from the different omics platform. It could be genomics, transcriptomics, proteomics, epigenomics, immunomics. So when the tumor tissue as such that we get from the clinical, the clinical sample is always very limiting. So you should try to extract as much as possible out of this tissue that is available to decipher what will be relevant for the patient. So if you have, so from one particular tumor sample, you can have a genomic approach, a transcriptomic, proteomics, epigenomics, immunomics, when all this data is generated. And then there is vertically, you're trying to align everything, integrate everything using different kind of tools. Then you are going to be having a multi-omics data. And this kind of integration will help in sample classification, in helping in disease subtyping. It can help in better biomarker discoveries. We will tend to understand the disease better, and then it can also help in prognosis prediction. And all this is influencing the era of precision medicine. So if you look at the key multi-omic modalities in cancer, what you do in genomics, we detect the mutations, we detect the copy number variations, the structural changes in DNA. And when you do a transcriptomics, one can look at the gene expression profiles, which will reveal all the active biological pathways which are operating in a particular cancer and the changes which are happening. Proteomics will tell you the protein abundance in the clinical sample along with its modifications, which will be reflecting the functional cellular states in the sample. Epigenomics can tell you the state of DNA methylation, the different histone modifications, the accessibility to the chromatin, which is in turn important for regulation of gene activity and which contributes to cancer progression. And metabolomics can talk about the different small molecule metabolites which are involved in altering the metabolic pathways. So all this can be studied in one particular platform. So little bit of basics about whole genome sequencing. What you do in whole genome, the DNA library is prepared through fragmentation of the extracted DNA, which could be from an FFPE tissue, or it could also be from a blood sample or a liquid biopsy, which is followed by amplification on the flow cell. The sequencing is done mostly on the Illumina platform with sequencing by synthesis, and then you generate a lot of data, which is aggregated in the form of a FASTQ file. And then this data is processed through a systematic bioinformatic pipeline, which will be aligned to the reference genome, which will result in the form of a BAM file. And from the BAM file, you will analyze your variants to give you the VCF file. So essentially what I'm trying to say is when you do these kind of multi-omic exercise, you're going to be generating wealth of data, a lot of data. What do you do in transcriptome? In transcriptome, you isolate the RNA from the tissue sample, then again you undergo a library preparation, and then you do a sequencing, and you're going to be generating a lot of data pertaining to differential expression. And then you again in RNA sequencing, now we have the single cell RNA sequencing, single cell DNA sequencing, single cell epigenomics, single cell proteomics, and metabolomics. So from the bulk tissue, now we are moving into the era of single cell. So that is the amount of precision that is coming up in the field. What do you do in proteomics? In proteomics, you can, when you're isolating the proteins out of your tissues or cells or a liquid biopsy, you can have two approaches. You can either have a gel-based approach or a non-gel-based approach. So if you're doing a gel-based approach, then you do a 2D gel electrophoresis, where you look at the differentially expressed proteins and then you cut them out from the gel, and then you characterize them, and then you look at you do a MALDI-TOF to understand what are the masses of those peptides, you identify the different proteins. But if you're doing a non-gel, then straight away you do an LCMS, and then you pick up the different protein markers. So these are very advanced methods, basically. So if you look at the advanced techniques in genomic sequencing, like gene screening, like the whole genome sequencing, one could do whole exome sequencing, and then you can generate wealth of data. And then you, as I mentioned, you have the single cell technology, which is providing new insights into gene activity. And then there are advancements in mass spectrometry, which is allowing accurate detection of vast number of proteins. All this progress has led to a growing emphasis on developing a data-driven computational and mathematical methods. Because when we are going to be generating a lot of data, you need a very high computational and mathematical capacity to understand and analyze this high-dimensional data sets from the various advanced analytical platforms. And we need to have a consistent molecular or clinical trait across the multiple data sets need to be derived. So therefore, this has to be, there has to be a proper way to integrate these diverse omics data sets that we get from the patients. And this is where the challenge lies. So as I mentioned, the rationale behind why is it that we need to do this kind of an exercise, how the different biological layers of the cells interact with each other. If you want to understand this, then this has to happen through an interconnected network, highlighting the underlying complexity of cancer. Cancer is a very highly complex disease. So when we are generating data from the different components, how we are going to be interconnecting this network, that is what is challenging. And the data integration in this case has basically three goals. Anything that we do has to be translational. So the three goals here is we need to understand the molecular mechanism of cancer. Why are we kind of indulging in multi-omics? Is because we want to understand the molecular mechanism of cancer. And then how these disease samples can be clustered, because it's all very highly heterogeneous. So we want to understand within this heterogeneity, how we are going to cluster the different disease samples. And then third, the most important, is it possible to predict the outcome? Is it possible to look at the survival or the therapy efficacy by this exercise? So this is the goal of the multi-omics. And then computational methods are needed to integrate this large diversity of data, prompting development of new algorithms to overcome the intricacies of multi-omics. So when we are representing and preparing the multi-omics data, this will involve very diverse type of biological information, because when you do genomics, transcriptomics, proteomics, metabolomics, epigenomics, the information that is going to come out of every single omic platform is going to be very diverse. So the challenge is how you're going to be integrating this diverse data. So how multi-omic helps? It helps in improving the cancer subtyping. It helps in biomarker discovery. Multi-omics approaches allow finer classification of cancer, identification of newer subtypes, and more accurate survival prediction. Like in the laboratory, we actually do for the early stage breast cancers, we do a finer classification based on the gene expression profiling by a particular test, which is called as the breast cancer 360, BC360 assay. So in one particular sample, we study like 800 genes, and this is helping us to classify them into different types. And that, based on a machine learning algorithm, we try to develop a risk of recurrence score. And based on this risk of recurrence score, we will be able to predict survival, whether who are the patients who have greater than 90% survival in 5 years or greater than 80% survival in 10 years. So these kind of approaches are possible. And when we integrate diverse data sets, we can pinpoint the driver mutations, what gene expression changes are important, what are the metabolic alterations which is linked to a cancer being aggressive, which can lead to an improved detection and prognostication. Second aspect is personalized medicine precision therapy. Gone are the days when we had like one size fits all phenomenon. Now it is all highly customized. So multi-omics analysis facilitates development of individualized treatment regimens, and it is also helping in dynamic intervention strategies. So if you look at the synthesis of data, including the liquid biopsy multi-omics, which is helping us in a big way, can help in early screening sensitivity above 92%. So therefore, a lot of sophisticated AI models, machine learning is helping us in these kind of exercises. And then the new technologies and the high throughput platforms, innovations within the multi-omics, you are having innovation, like you have the spatial multi-omics. As I mentioned, the single cell sequencing, integrated sequencing, mass spectrometry workflows, and ex vivo profiling platforms, all these are getting expanded into clinical implementation. Like I mentioned, I've given the example of the 𝐶IWA 360 that we do. And then also for identification of pathway and new drug discoveries. When you have a multi-omics integration happening, it will help in understanding, elucidating the mutation metabolism and immune cascades, which can help in driving the drug resistance and also to uncover targets for combination therapy. It will help us to identify new drugs which can be given. For example, the synthetic lethal strategies like usage of PARP inhibitors, ATR inhibitor combinations, because when we do profiling, you get multiple mutations. There'll be multiple mutations in the same sample. So we have to see what inhibitors will work, what will not work. So these kind of permutation and combination is possible these days because of the large amount of data which is available, and we are linking it with our results. So whether it is single cell or a bulk sequencing or a metagenome, essentially you're going to be generating a lot of data is what I'm depicted here. And then when you're going to be having a lot of data, you need to have high computational power. And this is where artificial intelligence is playing an important role. So as I mentioned, when you're going to be integrating the omic data sets, and what you're trying to do here is we are going to see whether there is a vertical causal relationship which is possible to be derived between different omic levels. And besides the same variant of the genome, why do we need to do this? Is because sometimes the same variant in the genome can cause different changes following levels due to the variant genetic and the environmental background. So it is highly heterogeneous. Therefore, application of multi-omics in cancer research will help in a predictable trend. So when you're going to be integrating, there is also a lot of challenges in these approaches. One big challenge is the data heterogeneity and integration. So when you're consolidating the multi-layer data from varied platforms, then it is highly complex because there will be differences in the data regulation, and especially the information may not be uniform. There may be sparsity of information when you're doing especially a single cell. It will not be uniform. So therefore, and as well as an uneven maturity across omics domains. So this will impede seamless integration. Therefore, this has to be taken care of. Second aspect is technical and computational barriers. All this data analysis requires advanced bioinformatics skills, and it needs very powerful computational resource. And most of the integration tools are also, they're continuously getting upgraded, and initially they'll be error prone, and then it is not possible for a non-specialist to use it. So one needs to have a strong bioinformatics or an AI professional or a data scientist working with the group to help us decipher the data properly. So therefore, the standardizing the data acquisition and processing is still a work in progress. And then when you're going to be involving a lot of patient data, then there is ethical, legal, and regulatory issues. So if you're trying to, come up with a clinical trial based on a finding, these clinical trials must address issues around the patient data consent. How is the data consent? What is the data privacy policy? So how this privacy is going to be preserved, and how this cross-border data alignment is going to happen. So there has to be a lot of regulation on these lines. And then there is also a need for interdisciplinary collaboration needs. It is not possible for just a clinician to do something in isolation. Now, whatever we need to do, we need to work as a collaborative effort, and that way we are very happy to work with ICGA here, because we from the hospital background, I'm a molecular oncologist, I have a team of clinicians with me, but then when we need bioinformaticians, we need data scientists. So all these collaboration is helping us to make us do something worthwhile. So therefore, the interdisciplinary collaboration is the way to go forward, and multi-omics is definitely stressing on this. So whenever there is a lot of data, how is it that we are able to link all this? Because there is a multi-omics database which is already available. Like you have with the expansion on acceleration of NGS, it has generated tremendous amount of data, isn't it? So you have the TCGA. The TCGA is the large largest pan-cancer multi-omics database, which has got a clinical annotation for allowing for any large meta-analysis. Anybody wants to do a large meta-analysis can get the data from the TCGA and do it. And now with ICGA also is trying to work on the same lines, where we are trying to generate data from our own country. The Indian data will get into the Indian Cancer Genomic Atlas, so that we will have something similar to TCGA in India. And then you have the Cancer Immunome Database, which is a new database out of immunogenic data and immunogenic analysis of NGS data from 20 different solid tumors, which have been derived from the TCGA. And then you have the International Cancer Genomic Consortium. This is again a database in place. Then you have the Molecular Taxonomy of Breast Cancer International Consortium, which is what is called as MetaBric. This contains genomic data from breast cancers, and it has helped in improving the classification and subtyping of breast tumors, which has already led to discovery of 10 different groups. And then you have the therapeutically applicable research to generate effective targets, treatments. This is called as the target. This target is specific for the pediatric tumors. So this data set empowers a description of driving process of childhood cancers. So this database is available. Then you have a Cancer Research Institute Atlas, CRI Atlas. This is a database platform which allows the study of interaction between tumors and immune microenvironment, which grants the possibility to explore immune response across genomics and clinical phenotypes. And then you have the iAtlas, which allows researchers to explore these readouts and relations. So there is a reference available. So if you see, these data sets can be used for any kind of an analysis. And then the recent advance in this era of multi-omics is oncoimmunology. So we are trying to understand the tumor biology and the immune together. So initially we were only looking at the cancer biology aspect. Now we are trying to see how the tumor cells are interacting with the immune component to see whether a tumor cell will be, whether it can respond to immunotherapy or not. So this area is picked up in a big way. So again, how you can derive data is same, your bulk sequencing as well as single cell sequencing. You can generate whatever data that you are generating out of it can be integrated using different tools and using different modules to understand how a particular tumor and the immune component interaction is happening. So if you look at this particular cartoon here, you are having a tissue. From the tissue, what is the information that you can derive? You can anyway look at the morphology. The morphology is from the histology. So when you look at a histological section from that particular tumor, you will be able to understand the morphology, and from that morphology, you will be able to extract features. Similarly, when you're going to be chopping the tissue into single cell levels, and then if you're going to be profiling them, then you will get the resolution of expression at the single cell level. And if you are going to be doing a bulk sequencing, then you will be able to get gene expression at the bulk level. So once you extract all the features based on this, whether it is a single cell references, it can deconvolute the spatial data, which can infer the cell type proportions and gene expression at spatial location. This, along with histology, can be coupled together to understand how the cell-cell interaction is taking place and what is the tumor-stromal interaction which is taking place. A lot of such information can be derived by this exercise. So as I mentioned, immunomics is picking up in a big way, and there are good tools available these days to study this. There is a package, R package for computational tool for immuno-oncology biological research. This has been developed to comprehensively combine and interpret multi-omics data in the context of immuno-oncology. And so this IOBR has been built easily to integrate whole exome RNA sequencing from bulk tumor as well as from the single cell RNA sequencing and long non-coding data. So it consists of like 4 module pipelines with a signature deconvolution for phenotype, for mutation, and for model construction. And this signature module can identify immune or tumor-specific signatures through expression profile and deconvolution estimate of the immune filtrate. Similarly, you also have certain panels and certain platforms. Like for example, you have the immune oncology panel in the mutation part is able to determine the association between. So as I mentioned, the integration strategies and technologies have to be highly robust here. So you need highly computational and mathematical methods are vital for combining this high dimensional omics data. And then you need to have advanced bioinformatics platforms like Galaxy, CBio Portal, which can process this multi-omics data, which will enable biomarker discovery and clinical translation. And also, most importantly, AI-driven tools, which can match the therapies to specific cancer profiles. So as I mentioned, it is going to be multimodal data sets, and they are all going to be stored in dedicated data structures that will allow efficiently to access and process. Each layer can be accessed independently or jointly. And then based on that, you can apply the machine learning integrated methods to decipher information. So why now, if you look at certain bioinformatics tools which are widely used for this purpose is like, for example, one of these tools they call for any kind of sample classification is a microarray logic analyzer. It's called as the MALA. This MALA is basically a logic data mining algorithm. So there is and it follows a three-step process. There is a discrete cluster analysis, and there is a selection of most relevant and cluster of genes, and then logical formulas which will characterize the samples. And then there is also another algorithm which is called as the joint and individual variation explained. It's called as the 𝐿IVE algorithm. So this tool aims to individualize two types of structures. You have the joint and the individual structure. Now, the joint structure is a biological pattern that you derive from the samples, which could be a gene expression or it could be an miRNA, whereas the latter is the intrinsic of one component that is unrelated to the others. And then there is also another diversity of tools which is based on matrix factorization, which is developed, which is called as the NMF, which is non-negative factorization. This consists of multiplication between columns and rows of matrix. So for example, if you have the data sets, so you have one matrix A, which is composed of, say, genes, number of genes that you have derived, and then the number of samples that you have used to derive those genes. So that is matrix A. And then the genes that regulate the expression of the downstream genes, the genes which are coming up significant, which will regulate the downstream genes, will be identified and they will be labeled as K. So this matrix A is split into two. That is, it is split into W and H. W is basically your N genes, the number of genes that you have got, and K, that is what the important genes which are going to be down-regulating or up-regulating the downstream gene, that will be K. And then you have the H, which is composed of K and M. So this W and H matrices are combined, and then this is used to form a new matrix. So these are all some of the ways in which these algorithms work. So if you see, these are innovative computational tools. These are all the different methods, the 𝐿IVE, the NMF. In the NMF, you have different types which have got their own pros and cons. And then there is another algorithm which is called as the lemon tree, which is something which they have used in a glioblastoma data set. So here, this is all an open code. It's an open source code which can be downloaded and used. Now, suppose if you have to use this particular multi-omic algorithm, you can define a biological question, like based on whether you want to understand what are the different copy number alterations in a lung cancer, which is relevant, say, co-express with a gene module that is coming up, or in any cancer, a glioblastoma or whatever. And then once you define your biological question, then you will pre-process the expression data matrix that you get, which can be downloaded if you are having it experimentally, fine, or if you are going to be getting it from a data set, fine. And then you infer the co-express gene clusters, and then you build the consensus modules of co-express genes. And then on the other way, this is based on the expression. On the other side, you will have the candidate, you select the candidate regulator genes, the ones which are important for gene expression or microRNA or copy number profile epigenetic profiles, and then you'll pre-process the input data, and both will get into an ensemble of regulatory programs for a set of co-expressed gene clusters, and then you can compute a consensus score based on that to see whether there's going to have a more recurrence, a poor prognosis, poor survival, good survival. Based on that, you can try to decipher the answers to the biological questions. So they have used this lemon tree software successfully to predict survival based on the certain genes which were relevant in the glioblastoma. And this is available, which can be tried. So machine learning is very, very important in this era of multi-omics, because the machine learning algorithms are the ones which are predicting models using statistical methods. So the algorithms here for machine learning can be trained in two ways. You can either have a supervised learning or an unsupervised learning. Supervised learning is based on a labeled input data set, which is used to train and develop a function to predict the outcome. So here the algorithm will attempt to find patterns which is relating to the features of the given label. And then the second step is to integrate the data, the test data, and classify the validation data in the right label following these identified patterns. And then you have to see whether there is a correlation between your given and the predicted label. This is what you do in a supervised learning. In an unsupervised learning, here it clusters the samples by identifying or regrouping the different features following a similar pattern. So if there is a similar pattern, it will group together. So therefore, the main difference in the unsupervised or the supervised is that in unsupervised, the input data will not be labeled. So therefore, and then you have a powerful tool for this multi-omic integration, which is the artificial neural networks. It's called as the ANN. This can be used for machine learning as well as for deep learning. Now, ANN is a very simple approach to create an artificial model, which is composed of neurons, which can be organized in layers. And then it is composed of an input layer, a hidden layer, and an output layer. So the input layer corresponds to the data set that you're feeding into the algorithm, and the output layer the output of your result is your output layer. And in this case, you will also have a hidden layer, which will define the complexity of the algorithm, and it represents possible ways linking your input and the output. Now, for the integration of multi-omics, the different blocks which are specific to each type of omics data are created. And then based on the different biological question, you can try and use different ML algorithms and see the ones which perform the best can be used. So there are vast diversity of algorithms which are available in machine learning. You have the random forest classifier, the k-nearest neighbor. All these are, commonly supervised classifiers which can be easily suited for a biomarker discovery. The random forest algorithm builds multiple decision trees that are randomly generated, and each decision tree will provide a classification, and the classification that is chosen in the majority of the cases is used to classify each data point of the data set. The k-nearest neighbor follows three main steps here. There is a reference data set, and it is clustered into three different classes that need to be distinguished. And then you have an experimental data that is integrated into the data set, and the experimental data is classified by calculating the distance of each of this defined class with the classifying data point in the nearest cluster. So these are all the different computational approaches which are available for multi-omics data integration. And then apart from that, you also have exploratory data analysis in multi-omics. So here we are going to have ways to reduce the dimensionality, because the data is going to be very diverse. The dimensions will be many. So therefore, you need to have ways to reduce this dimensionality. So there are dimensionality reduction techniques which are available, and principal component analysis, one of the very popular way to reduce the dimension. Then you have the t-distributed stochastic neighbor embedding, which is TSNE. Then you have the UMAP that we normally see in a single cell data. Most of the data is represented in the form of a UMAP, where the data gets projected into a Riemannian manifold. So all these EDA tools are a part of the bioconductor package. So anybody who's interested in the multi-omic area should be familiar with the bioconductor, different packages which are available as a part of this software. And then, these are some of the, each of this can be used. If you want to integrate transcriptome and proteome, then you can have this 𝐸𝐸𝐴D4, and then you have the multi-omics autoencoder integration. So these are some of the different tools which are, coming up with bioconductor, and one needs to play with this. So what you're actually achieving in this is, if you can do a network construction based on the data, you can have a network-based biomarker discovery is possible, you can derive the different pathways, differential network analysis, and then you have the the different dynamic network analysis, and then visualization of the data of the someone is talking. Hello,
Yeah.
I request you to mute, sir. So I will be spending a little bit of time on this. So this is what you exactly do. What you do in a multi-omic approach is, first thing is data collection. You will collect the data from various omics platform, which will ensure consistency in experimental condition and data format. So once you collect the data, there is data cleaning, where you will address the missing values, you remove the outliers, you add other data quality issues are addressed. And then after that, the data cleansing happens, then it is normalization. In normalization, you will do it across different omic layers, and then you will ensure that there is these data points are comparable, and which is very important for accurate integration. Once a normalization happens, and there is data transformation, where in this data transformation, this is done basically to achieve normal distribution and to scale the data, and this will improve. It's important to be done to improve the performance of the downstream statistical analysis. And after you do that, then you will do a feature selection. You will identify the relevant features which are important to give you information about a biological significance. And then you think of the different integration approaches, like it can be, and then how you're going to reduce the dimension, use, methods like CCA, PCA, factorization techniques. And after that, you're going to be annotating the data. This annotated data will then undergo data fusion, because the data fusion will fuse the integrated data into a form of cohesive representation, which will ensure information from the different omic layers is combined here. If it's proteomic, epigenomics, genomics, all the layers are combined here. Once it is combined, the next step is the visualization. You have to see how they look combined in the form of a heat map, in the form of a plot, in the form of dimensionality reduction methods. And then once you visualize, after the visualization, you will cluster them and subtype. So based on the common things can get clustered together, the uncommon things can get removed together. And once the cluster and subtyping happens, then you can apply your ML models to predict, outcomes or survival or whatever. And then whatever you have done, you're finding it using an ML model, that will be a test set. And that you have to validate on an independent data set. That is validation. And after that, you will give the interpretation of the context of whatever you have derived pertaining to a biological knowledge, pertaining to a functional relevance or a pathway. And then you have to again give it in the form of a visualization or heat map for people to understand. So this is the whole process of the multi-omics which happens, and then there are advantages and disadvantages. Advantages that it gives you comprehensive molecular profiling, which will give you a detailed analysis of multi-omic alteration in cancer cells. It can help you identify new biomarkers. It can unravel new molecular networks. It can improve subtyping, classification, identifying different molecular subtypes in a particular cancer type. Then it can help in enhanced drug discovery, target identification. It can help in understanding epigenetic modifications, which can help in early detection, screening, etc. But then, as I mentioned, the disadvantage is that it is highly computationally intensive process. So there is this temporal dynamics might be overlooked, which limits the understanding of molecular alterations. Then understanding the limitations of individual technologies is also crucial when you are trying to, bring them together to give you an accurate interpretation of the results. And often functional validation is required. So whatever you're deriving out of a multi-omic exercise, it will also need to be validated. There has to be a functional validation, and then the ethical and the legal issues with so much of patient data involved, and then high cost, because you are involving so many different high throughput technologies, and then you are bringing them all together. So therefore the cost of the entire exercise is also high. Summarizing, the integration of multi-omics approaches in cancer biology has revolutionized our understanding of the complex molecular landscape of cancer, and by simultaneously analyzing genomic, transcriptomic, epigenomic, proteomic, metabolomic, researchers have been able to gain unprecedented insights into intricate interplay of biological components that happens in cancer. And these comprehensive analysis have not only identified key molecular alterations which drive oncogenesis, but also have been able to reveal the heterogeneity present within the tumors. And then the depth of information obtained through multi-omic approaches enables identification of potential markers for early detection, for prognosis, for treatment response, also for helping effective clinical decision making. So to conclude, I'll just share a small bit of work that we did using an ML algorithm, something which is not fully, multi-omics to the scale that I talked about, but this is something which is relating ML and the clinical data, and where we tried to develop an XGBoost AI tool for predicting relapse in children with acute lymphoblastic leukemia. So this we have done on Indian population based. This is a study done on Indian pediatric patients. Why we did it is because these patients are treated on a uniform protocol. So the treatment is uniform. So therefore, the prediction of relapse can also be done on a particular application because the treatment is uniform. And this is based on the ICGA trial. So what we did here is, why we did it is because there are lack of predictive tools. Currently, there are no AI-based tools which can help to predict relapse in a pediatric ALL patients. So therefore, there is a significant gap in the personalized care. And then when we talked about it among the oncologists, there was a very high amount of demand. People wanted to know that if they could find out, before who are the patients who are at a higher risk of relapse, then that would help them to tailor treatment differently. So therefore, there was a high demand, and also to enhance the patient outcome. So what we did here is, we used an AI-based platform here, and then we have come up with the decision support. And what we have done is, we have built the classifier models, we have done the data validation and expansion, and we are trying to implement as well. So we used an AI-based tool called XGBoost algorithm. We tried different ML models, but an XGBoost performed the best to develop, and it was developed to predict relapse in ALL. The data was analyzed based on the ICGA pre-trial cohort, which had about 2,382 patients. And here we have integrated the clinical, demographic, and molecular features to provide personalized prediction. So we have looked at the different steps here. Was the, we did the iterative testing of models, we did feature engineering, we looked at the retrospective data analysis, we developed the AI model, then the model parameter was tuned, the performance was assessed, and we did a hyperparameter tuning, and then the model was validated. So these are all the different clinical factors which were, important features taken up to predict relapse. And what we found was, compared to all the different models that we tried, XGBoost's accuracy was, better than all the other models. So therefore, we took it forward. And then why XGBoost? Because these are all the different features of the XGBoost, because it is very efficient in handling missing data, because with a lot of patient data there, a lot of features will still be missed. So this particular ML model is very good in handling missing data. And then there is also an inbuilt cross-validation study. So these are all the features of the XGBoost. And we found that XGBoost performance was better compared to all the other. And this is the Prepare ALL, dashboard here, web application, where if you just enter the features here, then you will get the relapse score. The risk score is predicted here. And this is now we are going to be validating it on the trial data, the ICGA trial data. So therefore, I'm summarizing here, multi-omics has revolutionized our understanding. And then in summary, the application of multi-omics in cancer biology is ushered in a new era of precision medicine. It offers a more nuanced and comprehensive understanding of molecular intricacies of cancer. And there's going to be a lot of work which is going to be happening in this field. It holds the promise for further refining our knowledge, improving our diagnostic accuracy, and also enhancing her therapeutic strategies for benefit of cancer patients worldwide. So I like to acknowledge my the co-investigators for the Prepare ALL work, Dr. Raghunath Rangaswamy from IIT Madras, who's a data scientist, Subiksha Saravanan, who did the modeling, who used the different models and came up with the XGBoost, and Dr. Venkatraman, who's the clinician managing the pediatric ALL patients, and ICMR, which funded the project, and I'm the principal investigator of this project. I thank you all for your attention.
Thank you so much, Vijay, for such a wonderful talk and for all the wonderful work you are doing. This was really very impressive, and I hope the people in the audience would make sense of it and would like to adapt such methodologies in their, daily research that they take up. Thank you so much, Vijay. This was truly so good to have you.
Thank you.
Thank you, Vijay. I would now like to invite our second speaker for the day, Dr. Gagan D. Dr. Gagan completed his PhD in biotechnology at JNU New Delhi, and he received the DBT Wellcome Trust India Alliance Early Career Fellowship at the National Institute of Immunology. He has deep expertise in mass spec, proteomics, and metabolomics, and in 2017, he founded a laboratory which is now located in Noida that provides advanced mass spectroscopy services for research and diagnostic purposes. His lab is first in South Asia of its kind, which offers mass spectroscopy of such high resolution, and it demonstrates how cutting-edge tools can be translated into meaningful clinical applications. His talk today would be centered around industry perspective on multiomics and translational precision medicine. Welcome, Gagan, and the stage is yours now.
Thank you so much, Ankita, for the invitation, presenting our work here. So, Dr. Vijayalakshmi already has explained so well all the different aspects of multiomics and the latest IT tools available in the field, which people are applying to integrate the data. I will be focusing my talk more on the mass spectrometry side, in which our company having quite a good expertise in India. So a little bit about our laboratory, we started this company way back in 2017. 𝐕𝐼proteomics is our proteomics brand, and Valerian Advanced Diagnostic is our new, field which we are developing last 2 years, applying the mass spectrometry technology for advanced diagnostics, which give you much better quality data and foolproof data. So we have multiple orbit trap and triple quad systems in our lab. It's a very big 6,000 square feet lab space in our Noida facility, which is last year we have shifted. And we do all kind of proteomics, metabolomics, lipidomics, and lot of diagnostics in the new facility. We have a targeted panel NGS facility as well, laser microdissection labs. We do a lot of therapeutic drug monitoring, and we also do biomarker development with our academic partners and hospitals. Currently, the company is in a leadership position in the academic and clinical mass spectrometry research in India for development purpose. We do thousands of mass spec samples every year. And our mission right now is to introduce accurate and specialized mass spectrometry-based testing for diagnostics and care for our clinical partners. Brief snapshot what we do, right from sample prep for the clients till advanced bioinformatics, data analysis. These are the things which we do for our clients. We use, again, a lot of pipelines, open source, bioconductor, R programming, and many other kind of Jupyter notebooks, RStudio, Shiny applications for all this kind of work. Multiple collaborations we have, including our very good collaboration with ICGA for the proteomics for the breast cancer patients. We work with Institute Pasteur, Canadian University of Calgary, ISC Bangalore, AIMS. We have ongoing collaborations, IIT Kanpur, LB Prasad, NII, JNU, many other institutes not mentioned here. So if we talk about the basic concepts which we learned during our master's and PhD, central dogma of biology, DNA, RNA, proteins, and metabolites. And apart from that, many other kind of epigenomics technology Dr. Vijayalakshmi has introduced. So when you talk about all of them and you want to measure these molecules for your research system of interest, you call them multiomics. So DNA can tell you what can happen, and RNA gives you even much better data, what appears to be happening. DNA mutations are very, very important for so many kind of testing in oncology, more than RNA, I must say. And then proteins are further, I mean, the molecular machines which are giving function to the cell, and metabolites again are very, very important. So we target mainly on the proteins and metabolites, and once we find some biomarker related to the protein discovery, we further confirm them using NGS for some targeted panels using our thermo NGS machines. So basically, mass spec, it measure mass, and here is one advertisement from Google, you know, this, who put my lunch through the mass spectrometer, so you get spectra, masses. This is a very, very old slide, but all the mass spectrometer from any company right now in the market, they have this fundamental process by which they are building the mass spec. You have a sample which go into an inlet system, and then it go to the ion source. You have a very high vacuum inside the mass analyzer. And then after the analyzer, you have detector. Detector give you M by Z mass. The Z is the charge on the analyte of interest. Like in MALDI-TOF, you have +1 charge, and in ESI LC-MS, which can measure thousands of protein at one time, you have +2, 3, and +4 charges. And then you have a computer, both inside the mass spec as well as outside the mass spec, where you do the readout of the M by Z ratio. And then you have a lot of pumps around the mass spec, which create a very high vacuum inside the mass spec. There are different designs of mass spec available in the market. You can see here liquid chromatography on the top, then the traditional TOF, then you have 2 TOF detector TOF. Then you have triple quartz, which are very much used in the diagnostic field in the C section, as well as in the pharma industry. You have a MALDI sample plate on the right side, you can see. Then you have another variation of the mass spec hybrid one, in which you have quadrupole, and then you have a TOF attached to that in D. Then you have ion traps, and finally you have very, very high resolution superconducting magnet-based mass spec known as Fourier transform, FTMS, used in the nuclear industry as well as very special industry, because the resolution can go more than 1 million. So why measure the proteins? So there is a beautiful paper which came in 2012 from the Vogel group, and what they have done, they measure the mRNA abundance in a cell as well as the protein abundance in the same cell. And they have applied this study on multiple organisms. And what they could find, that protein abundances, they correlate much better between 2 species like C. elegans and Drosophila, rather than using only C. elegans, RNA abundance and protein abundance within the same species. That means there is a very low correlation between what is the RNA levels in any system compared to what are the real protein levels in the same cell. So there could be a lot of RNA which is just accumulating in the cell, not resulting into any protein. Or there may be so many proteins which are very abundant in the cell, but having very, very little RNA. So one has to be very careful when you are making conclusion from the NGS data. So RNA and metabolites, they give you much better picture of the functional aspects of any system. Application of mass spectrometry, there's so many: proteomics, metabolomics, lipidomics, clinical, which we are doing a lot. Pharmacokinetics we are doing with our clinical partners as well as drug development companies. We do a lot of plasma screening of their drugs. Clinical trials again, product developments, nuclear research, all the probes which are on the Mars or even in the universe these big NASA and all that they are sending, many of the detectors are based on mass only. Space research and again personalized medicine, which the earlier speaker also has talked about a lot. So proteomics are two kinds. One is a discovery proteomics and one is targeted proteomics. Discovery proteomics means you get a sample and you want to measure whatever kind of proteins are in the sample. And if you have biological replicate of that sample having multiple conditions, you run them on the machine one after another. And then you measure these samples and then you find the ratio which are changing for the identified proteins between the samples. And you come to a relative ratio like fold change, which will tell you that which proteins are upregulating or downregulating or uniquely expressing in your which sample control and treatment sample. This is also known as shotgun proteomics, and it gives you an exploratory kind of investigation. So suppose you have cancer tissue patient samples from non-cancerous tissue as well as cancerous tissue. So you can have 10 non-cancerous sample and 10 cancerous sample, and you can make 20 sample, run them one after another in a discovery proteomics method, and you identify thousands of protein, and then you can put them in a ratio manner, and you can find the fold changes. Once you find the biomarkers which are highly upregulating or downregulating in your current research system, you can apply targeted proteomics. So suppose you find 3 biomarker A, B, and C, then you can design special spiking peptides, or you can design an in-house kit for targeted proteomics, and you can measure exactly in femtomoles or ppb or ppm your protein present per ml of your lysate. So that is a targeted proteomics. It is a hypothesis-driven investigation, and it required very different skills compared to discovery proteomics. The traditional way of doing proteomics was you run your lysate on a 2D gel. The gel is run 2 times based on PI and molecular weight, and then you cut the protein spots. Generally, people were able to identify 400 or 500 protein spots in a very good run 2D gel, and then you cut these spots in the untreated and supposed treated condition and do a MALDI-TOF. So that was the way of doing traditional proteomics. And here it is showing that once you get your MALDI-TOF sample ready, you can put it on a laser plate. The laser will come, laser will ionize your peptides. The peptides will be search against a search engine like Mascot, which will match the theoretical spectra with your experimental spectra coming from the MALDI, and it will give you finally a list of the identified protein or proteins coming from the gel band. Later on, after 2000, there was a big revolution for ESI-MS-MS, which is using nano columns or micro columns for detecting thousands of proteins. So today the technology has reached to that stage where the depth of proteome which you can cover by a very good prepared proteomic sample is as close to NGS. People are routinely identifying up to 10,000 to 11,000 proteins in the proteomic samples. In NGS, people are identifying maybe 12,000 to 13,000 transcripts, suppose for the HeLa cell lines, but you can also detect similar number of proteins using very high-tech systems which are available today. In any kind of nano ESI, LC, MS, MS, the basic principle is chromatography followed by mass spectrometry. So here you can see that there is a 80-minute mass spec run in which you provide a acetonitrile gradient. So the hydrophilic peptides, they will be eluted from the HPLC very early, and hydrophobic peptides, they will be eluting very late. And they will enter the mass spec one by one in a form of a nano spray, which you can see here, the Taylor cone or the plume. And the machine, based on the speed of the machine, will keep sequencing these groups of peptides which are entering the mass spec. So what happened in most of these machines, either from Thermo or Bruker or any other company or Zyx, you have a MS spectra in which you have a mixture of group of protein entering the machine. As you can see here in the A diagram, you have multiple peptides going as a survey scan inside the machine, and then the machine having an inbuilt computer which does a data-dependent acquisition. So what it will do, it will tell the gatekeeper, the quadrupole of the machine, to select these molecules one by one, these peptides, in a pure population, and do the sequencing of this pure population one by one. Now, the sequencing of the peptide is known as MS-MS, and the intact peptide is known as MS. So you can see here the A peptide mixture is sequenced again and again on the B section, different masses like 227, 255, 281, 283. These are different peptides. So in this way, the today machines using very high resolution and in-depth analysis can identify thousands and thousands of protein in any tumor sample, any cell line. And how mass spectrometry can be quantitative. So what the machine computer do, when it read any peptide entering the machine, it start making this sigmoid shape or bell-shaped curves for the peptides which are entering the machine. So what people do, they measure the chromatographic peak area under the curve, we call it. And this area under the curve is used for quantitative proteomics. Suppose you have 10 sample. So the machine algorithms, they will align the 10 sample, and then they will align the different areas of the peptide detected in the 10 sample, and you identify the fold changes based on the area calculation. There's so many quantitative MS technology. If you go to the literature, and people sometimes they get confused that which is the right technology from them. In our laboratory, we are extensively using label-free, which is also used in 99% of the labs all around the world. But there are many other commercial technologies like TMT, SILAC, which are quite expensive and kit-based. Very, very few labs are today using the kit-based technologies, but most of the labs, they are using label-free, which we also use. So basically, all of these different technology which we have written above, they are based on only two concepts. Either the peptide is measured at the MS1 level, or it is measured at the sequencing level, MS2 level. Any kind of mass spectrometry experiment, whether you want to do proteomics, metabolomics, or lipidomics, this is the workflow which people are following. You have a cohort, like you can see here, the human cohort. You extract the sample, you process the sample, you make peptides, or you make metabolites, or you make lipids out of it. You run it on a LC. The LC is connected via a column to a mass spec. And in the mass spec, you can use different technology like DDA, DIA, PRM, or discovery or targeted, which we just discussed some slides back. Once you get the machine raw file, the raw file is searched against a search engine. There are so many search engines available from different vendors as well as open source. After running it on a search engine, you get a list of protein or peptides or metabolites or lipids. And then you apply different statistical testing. Once you get a statistically significant proteins, you can do a lot of downstream analysis on it. Like you can generate heat maps, volcano plot, PCA plots, correlation plots. The earlier speaker talked about the detail, much more detail, the things which can be done with the list of proteins or list of transcripts which you detect in your NGS experiment. You can further do enrichment analysis of these pathways, and you can also apply machine learning and AI to further refine your data. And you can even find those patterns which cannot be found by traditional way of data analysis, which is mentioned here. And further, once you find your biomarkers, you can further validate them using, again, targeted proteomics or targeted metabolomics, or one can also use immunohistochemistry, or they can do Western blotting or flow cytometry if those tools are highly refined. So what we do in our laboratory, we have a lot of in-house developed optimized protocol for sample preps. We do a lot of customized hardware for our machines, which we are using in our lab. We are not totally dependent on vendors. For the mass spec, we develop a lot of tools in our lab. We have many kind of automation robots in our lab for large sample processing, big projects. to even identify things using our machines today. So using the latest technologies which are available for the mass spectrometry, you can get much better quantitation and coverage of the proteome. NGS, they generally identify 11,000 proteins, 11,000 gene expression, using a 20,000 reference human database, and our proteome also is reaching nearly 72% of the NGS coverage. One example of the latest work which we have published, it was published in Cell using phosphoproteomics. There are not very great papers from India in phosphoproteomics. So we have a very good collaboration going on with IIT Kanpur with Dr. Arun Shukla, and he is working on G proteins, which are very hot targets for drug discovery. So we have already published many papers along with Dr. Shukla, and in this paper, we have applied very advanced phosphoproteomics enrichment methodology, which are our in-house methods for phospho-enrichment, followed by a very advanced bioinformatic analysis. So using this technology, we are currently applying the similar phospho approach on plasma sample from Alzheimer and Parkinson sample to find phosphoproteins which may be changing in neurodegenerative pathways. So phosphoproteome regulation in biological or models of biopsy tissues, they can be of very much interest to find lead candidates for biomarker discovery, but you need very highly standardized and reproducible enrichment and good technical expertise to do phosphoproteomics. And one example is p-tau assay, which is approved by US FDA, and Roche and other companies, they have launched it. And many American companies are already working on it using mass spec for Alzheimer's disease detection 10 years before any Alzheimer's symptoms are appearing in the patient. We are also working on similar technology using multiple biomarkers with our own machine learning models. One more example I can very good results. We have done this project with Dr. Puneet Kaur. More than 3,000 samples were analyzed for multi-drug resistant bacteria, E. coli, Salmonella, Pseudomonas, and Klebsiella, and label-free proteomics, as you can see here in the heat map, can easily identify the MDR multi-drug resistant bacteria from non-MDR multi-drug resistant bacteria. Similarly, on these kind of databases and data points, once you generate, you can apply AI/ML and you can make mathematical models for the prediction. One of the things which we take very much proud in our Noida lab, we are the first lab in India, in fact, in South Asia, which has launched amyloid typing. And there is no other lab after our lab, even in Japan and China, which can do these kind of testing. So there are two labs in the world. One is Mayo Clinic, the other one is UK NHS, which have the similar capability. So amyloid typing we launched first time in South Asia, and we are receiving amyloid sample from all across India. This is our lab picture. We have two laser micro dissection microscopes. One is from Leica, one is from Zeiss, and we have multiple orbit trap systems to find the amyloid biomarkers. We are applying the similar technology using laser micro dissection on formalin fixed tissue for cancer tissue profiling, as well as kidney membranous nephropathy, for which there is only one lab in the world, which is Mayo Clinic. So amyloid typing we have done with very much expert guidance with National Amyloidosis Center, UK NHS, Dr. Ashutosh Vachirkar and his team, and lot of amyloid sample we have done along with Dr. Lal Path Lab. Dr. Alok Sharma is our contact for the same. So this commercial testing we have started in India first time since May 2025. In our lab, we use a lot of automation, automatic robots for sample processing for both metabolomics as well as proteomics, as you can see here. And these remove the inconsistency of the data of the sample preps. We also do a lot of therapeutic drug monitoring using mass spectrometry, using our triple quad platforms. We have a lab in Saket, and it's a fully operational lab since 2023. And we are nearly doing 3,000 patient immunosuppressant testing every month using mass spectrometry. So this is one very good example of mass spectrometry, which can give you very accurate data compared to CLIA or ELISA. We recently started monoclonal antibodies measurement for IBD, inflammatory bovine disorder patients. And we again received sample all across India, especially for infliximab and adalimumab. These are two monoclonal antibody for IBD patients. So there are very clear-cut guidelines available for these two monoclonal antibodies, that how you should use them in the proactive and the reactive therapeutic drug monitoring. So we are getting patient sample both from proactive and reactive patients. Generally, most of our samples are coming from reactive therapeutic drug monitoring, because many patients, when they are under a clinician, they are not responding well to the biologics. So clinician really wanted to know that what are the levels of the biologic in the patient sample. And many cases we are finding that the level of the biologic is very low, which is as per the trough level doses expected from the treatment. So in these cases, we have again using mass spectrometry, we developed very accurate gold standard methods for anti-drug antibody measurement against the biologics. So not only we can measure the real biologics level in the plasma sample, we can also measure if there is any anti-drug antibody made by the patient against the biologic, because of which they are not responding well to the treatment. If these kind of decisions has to be made, and if a clinician find that there is anti-drug antibody against the biologic, then the physician has to change the therapy. They have to either change the biologic or they have to bring the patient to another drug. So far these technologies were not available, but Valerian, being a pioneer in the field of mass spectrometry, is bringing this technology for different biologics in the field. One example I can give you, like 4 different monoclonal antibody drugs like infliximab, adalimumab, vedolizumab, and ustekinumab. These are the induction phase, post-induction, and maintenance. So the maintenance basically means the patient has to have 5 to 10, or suppose 8 to 12, or 1 to 3 of usteki, inflixi, and adali monoclonal antibody after the week 14, 12, or week 14 or to 8. So we can measure the biologic concentration at any stage, induction, post-induction, and maintenance. And there are guidelines available for many kind of diseases, clear-cut guidelines where it is recommended to measure the biologic levels in the treatment patients. For the cancer, guidelines are still under progress, and we hope that in the next 2 to 3 years, lot of data will come from big groups in which biologic concentration measurement for cancer therapy will become compulsory. One more example which we are doing right now in our lab, in the left panel, you can see that these are each different monoclonal antibody peaks. So we have developed very special methods like 10-minute methods in which we can measure multiple biologic at the same time. So in traditional ELISA, for every biologic, you have a different ELISA plate and it takes a lot of time. Our technology can measure 10 to 12 monoclonal antibody at the same time in a single run. So it doesn't matter the patient is on which brand, the patient is on which monoclonal antibody therapy. Our methods remain same since morning to evening when our machine is running. So we can measure any of these oncology segment monoclonal antibody in our machine, rituximab or nivolumab or ipilimumab. It doesn't matter. Even if we know, even if the physician don't tell us which biologic the patient is going therapy, we can still tell it. And sometimes we have observed that if the biologic therapy has been changed, some lingering amount of previous monoclonal antibody because of the treatment 6 months earlier, which was given to the patient, still we detect it in our mass spec with the latest biologic as well as the previous biologic. So we have developed these kind of different kind of segments for oncology, inflammation, as well as anti-drug antibody for infliximab, adalimumab, ustekinumab, and vedolizumab. And we are the first lab in the country doing this by mass spectrometry. We provide the results same day or the next day with our gold standard MS technology from our Noida lab. The conclusion is mass spectrometry is the unbiased and gold standard technology for accurate measurement of analytes, both for R&D as well as clinical diagnostics. And it's a very rapidly developing technology. It also requires a lot of computational power, very latest AI ML tools. And today, with the latest technologies, Dept is coming very close to the NGS platform, but you get much more meaningful data. A study of protein, lipid, and metabolite, as per the central dogma, they give you much better understanding of the information of the cellular system compared to DNA and RNA analysis. NGS analysis has low correlation with real protein abundance, so one has to be careful. Especially the PTM modification of proteins is another frontier, which is very, very interesting for neurodegenerative as well as many other disorders. And targeted mass spectrometry technology has the capability to replace many ELISA-based methods, which are existing in the market and giving false results because of the cross-reaction of the antibodies. Thank you very much.
Thank you so much, Gagan, for the deep insight into MSMS. And this was really wonderful to know the kind of work you are doing and offering such deeper diagnostic services to the clinicians and helping in timely management of the disease. This is wonderful, actually. I now open the floor for questions, and audiences, please feel free to share them in the chat box, your questions. So in the meantime, I would like to ask one question both to Vijay and Gagan, that what are the biggest barriers you feel in the way of scaling multi-omics adoption in India? Is it the cost, the data interpretation technologies, the infrastructure, or we have less people trained for doing so?
Can I answer that?
Sure, Vijay, go ahead.
Yeah, I think all the factors are important.
Yes.
All the factors play. Like, for example, in my case, I may be strong in genomics, but I have zero knowledge in proteomics. So we have a person like Dr. Gagan, and we should collaborate. We should have, that is why this multi-omics, the best thing is it is bringing the collaboration in a big way. And then, yes, cost is definitely a concern because, we are always limited with the amount of funds we get for a grant, or all these things that we are trying to develop. A concept is based on the grant. So in the grant, we will never have one grant will not be enough to do all this. And we have gone through this, Ankita, together when we have tried to apply. So I guess so there has to be separate funding agencies for this multi-omics. People should encourage multi-omics to be in place because the patient sample is so limiting and so much we can get in one test, and if we collaborate and do it. So I think all the factors are important. Thank you.
Yeah. Gagan, your take on it?
I think she has answered everything very well. I don't have much to say. Yeah, I mean, collaboration is the way to go about it. And we are very, very thankful to the clinicians which we are working, and they give us such a good cohort and great samples and
Absolutely.
collaboration. And also, it's very important to create I believe like AI and machine learning, it will only work if the quality of the raw data is wonderful, coming from NGS or even for metabolomics or proteomics. The quality of data has to be very high class, the experimental variations. And that's why we apply robotics now in our sample prep. I mean, our people are excellent. I have no doubt about it, but sometimes, robotics, they help a lot. Yeah.
No, I'm very happy to see such good work happening in India pertaining to proteomics, because we have been looking for partners to work on proteomics, plenty of clinical samples available with us. And I'm coming to you, Dr. Gagan, because there is so much to be done. Thanks, Ankita, for this forum. I've got to know Dr. Gagan. because this is an area where we need good proteomics. We have done some proteomics work, but we have not been able to get very good quality data out of that. We did 2D and we have been able to derive some markers. But as you said, there is a lot of difference between what you derive in NGS and on the proteomics. They are two different things. So it's better to work on the protein compared to, working on the things. So
Actually,
therefore,
NGS is amazing in the way, in proteomics, we are using the traditional UniProt databases, which are like 20,000 for the human. But we know that there are so many isoforms and mutations.
yeah.
So for the same cancer tissue, if you have the NGS data and you can create your own
Yes.
library,
Yes.
if you apply that in proteomics, you get very, very novel discoveries.
Yeah, yeah.
So that's why the marriage of both NGS,
It's
genomics, and
definitely
proteomics has
important.
to be together.
Yes,
Yeah.
yes. So I think great initiative by ICGA, because ICGA is working on the multi-omics
Yeah.
area in a big way, bringing the experts together on one platform. So that's a great initiative, and I'm sure good data from India is going to be coming out.
That's what we are looking to and trying to create with help from you and Gagan and people across India who are equally, motivated to get something done for the betterment of our people, So I hope in few years we'll see that happening and things would mature to that grade where we can, specify and develop things for our own people. Yeah. So there is one question in the chat box that are there any examples of institutional initiatives of multi-omics in cancer?
I think there is. That is what we are trying to do, Ankita.
Yes, absolutely. Yes, absolutely. So a lot of hospitals across India are now integrating NGS and multi-omics into identification of patient subtypes, and they are giving tailored treatment according to the profiles that they display. Yeah. So as I think there are no more questions, so we'll wrap up today's session. And I would together, both the talks today underlined that multi-omics is not just a vision for the future, it's already a reality that is steadily shaping the present state of cancer care in India. And on behalf of ICGA Foundation, ECO Platform and CHIP Foundation, I thank our esteemed speakers, our participants, and the organizing team. Please stay tuned for the upcoming episodes in our next webinar series. Over to you, Shekhar.
Well,
Dr. Shekhar, are you around? Yeah.
It has indeed been a great session and a lot. Yeah, I hope everyone. It has been a great session with Dr. Gagan and very passionately, and we have all heard about so much about the economic aspects and about the clinical precision of proteomics and genomics. It has been really a great session. We had a good number of participation as well from the participants, and I hope the in this cohort will now take it up as research topics in this proteomics field, and bring out with some concrete scientific evidence so that cancer management can be planned better. So we getting this platform to thank our respected panelists and as well as this webinar. So that's it for today, and thank you everyone for joining us in this session. Thank you, everyone.
Thank you so much, everyone. Good day. Yes, I'm there. Yeah.
Thank you all.
Thank you, everyone. Good day. Bye-bye.